Thank you so much to the organisers. It's such a pleasure to be here. So today I'm going to speak to you about the impact of heartbeats on cognition and emotion. So the body and brain are intrinsically and dynamically coupled. Our thoughts, feelings, and perceptions are shaped by internal bodily signals, such as heartbeats. And so extraceptor senses, such as vision, touch, audition, help us navigate the world. And in contrast, we have interoception, which is an internal sense, the sensing of internal bodily signals. And it's a relatively new sense, and it's been somewhat controversial about how we best define it. So there was an interoception conference that brought together 100 researchers, and we came up with this shared definition in a white paper. Interoception refers to the process by which the nervous system senses, interprets, and integrates signals originating from within the body, providing a moment-by-moment mapping of the body's internal landscape across conscious and unconscious levels. And the organ that I specifically focus on is the heart. And much of my work has attempted to delineate different dimensions of interoception across these different hierarchical levels. So we have the nature of the afferent signals themselves, such as heartbeats. We have the neural processing of these signals. We have the preconscious impact of these signals, that is, how the signals change the way that stimuli are processed. We have the accuracy with which people can detect these signals. We have self-report and beliefs that people have about these signals. And we have insight, because people's beliefs don't necessarily match onto what's actually happening. So an insight's a measure of how good people think they are relative to their ability. People also differ a lot in their attention to these signals and also the attribution of these signals. So much of my work delineates these different levels and different clinical populations to look at selective levels of disturbance. And for the purpose of this talk, I'm going to mainly focus on these lower levels, as there's greater analog for animal models and animal studies. And first of all, I'm going to look at the nature of afferent signals. So in this study of nearly 400 patients, we looked at heart rate and heart rate variability across all these different patient categories. So healthy controls are in green. Then we have depression, anxiety, mixed depression, anxious, bipolar, emotionally unstable, personality disorder, also known as borderline, schizoaffective, and schizophrenia. And what you can see is that the healthy controls have the lowest heart rate. And in all of these different mental health conditions, there's elevated heart rate, with it manifesting as highest in schizophrenia. And in terms of heart rate variability, you can see the highest level of variability is in the controls, and then you get this sort of monotonic decline. The lowest is in schizophrenia, and I'm so fascinated by the fact that schizophrenia is associated with this very, very regular beating heart. And this replicates work that's been done by many labs across the world, that you get these interesting differences in these cardiac signals and patterns. And they also do seem to be informative for the condition itself. So there's work done in a Scandinavian cohort, a longitudinal sample, that looks at heart rate in healthy young men aged 17 and 18 who are being screened to join the army. They're not showing any signs of schizophrenia, but a high resting heart rate, specifically over 80, actually goes on to predict a greater likelihood of getting schizophrenia. So you get a change in these afferent signals before symptoms of the disorder manifest. And then we also can look at the neural representation of these cardiac afferent signals. This can be done in a number of different ways. I'm just going to highlight one way quickly now, and I know we're going to have talk on this later on. So we can also look at the heartbeat evoked potential, the HEP, which we can do using EEG, where we look at the cortical processing of the heartbeat signal. And we can see that in individuals such as those with borderline personality disorder, there's a reduction in this HEP signal. And the amplitude of this signal, you can see in this graph, is lowest in individuals with borderline, medium in individuals who are borderline but in remission, and it's highest in healthy volunteers. My own work, which is led by a neurologist called Mahinda Yogaraja, is interested in the HEP and its role in functional seizures. Now, functional seizures look just like epileptic seizures when they actually happen. So in terms of the behavior itself just looks like an epileptic seizure, but you don't get classic epileptic-like activity in the brain in these individuals with functional seizures. So it's been a sort of mystery. These individuals get passed between neurology and psychiatry because they can't find a clear origin in neural activity. But what we can see by looking at this measure, which pertains to body-brain integration, specifically this heartbeat evoked potential thing, is that the amplitude of this signal is actually predictive of a seizure. So interictal means just between seizures, and the amplitude of the heartbeat evoked potential, so basically at rest, is equivalent between our epileptic group and our functional seizure group. And then pre-ictal, which is just before a seizure happens, you can see a marked drop in the amplitude of this heartbeat evoked potential signal in the brain that predicts the onset of a functional seizure. So looking at these measures of body-brain integration can also be informative for clinical conditions. And next, I want to talk about the pre-conscious impact of the afferent signals. This is how stimulus processing is actually altered by the nature of the signals in the body. So every time our heart beats, it sends a signal to the brain, so our brain is activating dynamically in time with each heartbeat. And this happens via baroreceptors, which are mechanoreceptors, which are pressure-sensitive receptors, which are actually activated by the cardiac ejection period. And these send signals to the brainstem and brain via the glossopharyngeal nerve and the vagus nerve. This talk is going to focus on the functional implications of this signal, but for those that are interested in the neurocircuitry underlying the effect, I highly recommend this paper. So we can look at the functional impact of these cardiac signals and their interaction with brain by doing experiments which specifically time the presentation of stimuli to coincide with when the heart-brain channel is active. That is when the heart is signaling to the brain the degree of cardiovascular arousal. And that actually happens here at T-wave, which is the cardiac ejection period. And it's actually at this point the heart and the brain are in active communication. I'm going to also refer to this as systole for short. And in contrast, you have late diastole, which can be seen at R-wave, which is actually in between heartbeats, where this heart-brain channel is quiet. And you can time brief stimuli to arrive here or here to see how this active heart-brain channel changes the way that stimuli are processed. And it's been thought for a long time that these cardiac afferent signals actually have an interfering effect or an inhibitory effect on how stimuli are processed. And the early work that investigated this effect primarily used sensory stimuli, such as pain. And my work was interested in seeing whether we could also have higher order types of processing also impacted by these cardiovascular signals. So in a surprise experiment, it was a memory one, but I didn't know it was a memory one. What I did was I timed the presentation of words, which they thought was an attention experiment, to either T-wave or R-wave, so systole or diastole. And then I gave them a surprise memory experiment at the end, just free recall, name all the words that you remember seeing. And those words that were encoded at systole were systematically less remembered than words that were encoded at diastole. So there was an interference effect on the consolidation of these words if they happened exactly when the heart-brain channel was active. And then wanting to see if we could further extrapolate the inhibitory or interfering effect of heartbeats on other types of processing, we did a virtual reality driving experiment, which used this sort of steering wheel and pedals in a VR setup. So it was actually super realistic. We did it on 60 participants, and we had people drive, but we had obstacles appear on the road. And these obstacles were time-locked at different points in the cardiac cycle, either R wave or T wave. So these are the different types of obstacles we had, and we also manipulated the speed of these obstacles. So did they come fast or slow, and were they appearing near or far? And I'll show you what this looked like. So this is actually someone doing the experiment. They're reversing, and now they're driving. There's a barrel. They swerve to miss the barrel. There's a child. They hit the child. I'm really sorry. And there's another child, and they avoid the child. So there you can see that. And so what we can see, and this is like messy data. I mean, it was like a computer game, but you can see that there is a main effect of speed in that obstacles that were appearing fast were they had a longer reaction time relative to those that are slow. Each dot is a person. So you can see the general tendency to be slower for these fast-appearing stimuli. For near relative to far, people were again slower. For the near relative to the far-appearing stimuli, and we have a comparable effect size for cardiac cycle where individuals were impaired in their capacity to react to stimuli which appeared at systole relative to diastole. So again, these cardiac afferent signals are interfering with the processing and reaction to these stimuli. And coming back to this early sensory work, so much of this early work was done on pain. So here, pain stimuli were time-locked either to R or T wave, and you can see that the nociceptive reflex to these pain stimuli are also dampened down if a pain stimulus happened exactly at cardiac systole. So this shows that when cardiovascular arousal is represented in brain, there's this dampened pain response. And what this is actually showing is it's actually insight into the system of when brain receives high cardiovascular arousal, the subsequent effect on pain. And I think the point I want to make is, yes, this does happen on a moment-to-moment basis, but it also has implications for what happens when your whole body is in a state of high cardiovascular arousal, such as under an extreme stress response, which can happen also, of course, at longer timescales. So this effect can also be extrapolated to, for example, times of heightened stress. So to look at this in a different way, we work together with Flow, which is a period tracking app. It has over 40 million users worldwide, and we were able to get data on 100,000 Ukrainian women. And we were able to look at pain and stress, and we can see that pains are represented here, stress is here, and these are all other different types of symptoms, such as mood and other things. And exactly the point that the war and invasion occurred, you can see there's a rapid spike in people's reported stress and a corresponding dip in people's reported pain, which is very much in accordance with this principle that cardiovascular arousal can actually have a dampening effect on pain. And in this analysis, looking at the degree to which stress is impacting pain and looking at the size of this effect, you can see it's heightened here in Ukraine as well. So much of this work points to dampening down of stimulus processing with these cardiac afferent signals. Memory can be impaired, pain can be reduced, reaction time can be slower, and this is very much in line with the Lacey and Lacey hypothesis that these signals are always inhibitory or interfering. And I wondered whether this was universally true, which was what was supposed at the time. And much of my work had been with people with post-traumatic stress disorder, where I was trying to understand fear circuitry in the brain in these individuals, and was struck by the level of sympathetic arousal in the body. And I had wondered actually how this heightened bodily sympathetic signals might be driving increased fear memories and fear processing. So I wanted to see whether these cardiac afferent signals could actually boost fear as opposed to dampening down fear, which is what you see in most other categories of processing. So in this initial study, I just time-locked fear phases or neutral phases to different points in the cardiac cycle and got people to rate how intense this phase was, and this happened in the scanner. And what we could see that actually when the heart-brain channel was active, you actually get an increase in fear intensity ratings. So the fear is actually boosted with the cortical processing of these heartbeats. And in contrast and in line with the other findings, you get this dampening down of neutral intensity ratings with this cardiovascular signal. And in brain, you can see a main effect, a cardiac cycle, whereas amygdala actually tracked the cardiac modulation of fear, with increased amygdala activity at systole relative to diastole for fear stimuli, and the reverse was shown for neutral stimuli. So seeing whether we could extrapolate that and apply it to a fear conditioning paradigm, we time-locked a CS plus and a CS minus to diastole, and a CS plus and a CS minus to systole, with 70% shock following the presentation of the CS+, and we only analysed trials in the absence of shock, so it wasn't contaminated. And these stimuli are only presented very, very briefly, so they hit that part of the cardiac cycle. And we have the specific hypothesis that if cardiac systole does indeed boost fear, then actually you should see an increased fear response for both the CS plus and the CS minus for stimuli that are time-locked to cardiac systole. And we looked at skin conductance response as a measure of the fear. So in the initial trials that we analyzed, that's exactly what we found. So we saw an increase in the skin conductance response for both the CS plus and the CS minus, which were time-locked to cardiac systole, and a reduced response for both the CS plus and CS minus that were time-locked to diastole. And breaking this effect down, we actually found that this was driven by those individuals who were more anxious. So for the low anxiety group, there was equivalent collapsed over CS plus and CS minus for systole relative to diastole, whereas in a high anxious group, the elevated systole response was manifesting there exclusively. And for those who are interested in just seeing the individual data points, it worked, yes, both as a medium split, but also as a correlation, as a function of individual differences in trait anxiety. So those individuals who were more anxious were more prone to have this exaggerated fear response to a stimulus presented at systole when the heart-brain channel was active relative to diastole, irrespective of whether it was a CS plus or CS minus. So in the interest of time, I'll just let you know that late fear conditioning, there was no difference in, there was a significant difference in CS plus relative to CS minus, thus fear conditioning effectively worked. And by the end, there was no effect to the cardiac cycle. The next day, we then followed immediate extinction here, and then brought them back, and extinction was effective, and we brought them back the next day when we looked at a reinstatement procedure. So we gave them random electric shocks, just 4 presented, and then we showed them the stimuli, CS plus and CS minus again, and this time they were not time-locked to the cardiac cycle. And here we saw an exaggerated skin conductance response to both the CS plus and a CS minus that were previously presented at systole relative to diastole. So this exaggerated fear memory for stimuli processed in time with a heartbeat manifested the next day following this reinstatement procedure. So to conclude, the brain and body are intrinsically and dynamically coupled. We can see different patterns of heart rate and heart rate variability in different clinical populations. And as well as looking at brain activity independent of organs, looking at how organs are processed by the brain can also be an informative measure that maps onto symptoms and differs in different clinical populations. And cardiac signals can change extraceptive and interoceptive processing to shape pain, fear, and memory. To conclude, thank you so much to my collaborators, my lab, the Research Council, and thank you all for listening.